A warm and enthusiastic welcome to all of you on this esteemed platform dedicated to scientific talk in the field of computational biology and chemistry application in the medicinal chemistry. We all know that medicinal chemistry stands at the forefront of biomedical research, where groundbreaking discoveries have power to revolutionize the way we treat the diseases and alleviate the human suffering. Today's talk will cover a wide array of topics, ranging from your latest advancements in the drug discovery and development to novel therapeutic targets and cutting edge technologies that's going to drive in the medicinal chemistry research. With this, I am delighted to introduce our esteemed speaker, Dr. B.R. Prashant Kumar Sir, M.Pharm PhD, Associate Professor in the Department of Pharmaceutical Chemistry, JSS College of Mysuru. He is a prominent researcher and expert in the field of glitazones, targeting for PPR gamma and alpha receptors. Over the past 16 years, Dr. B.R. Prashant Kumar Sir is dedicated his career to pioneering a research focused on the development of novel glitazones. With a multidisciplinary approach, Sir is compensating computer-aided drug designing, synthesis, analysis, and screening. As a testament to its impactful contributions, Sir has have impressive track record of 72 research papers published in prestigious peer-reviewed journal along with two granted patents. Beyond his scientific accomplishments, he is deeply committed to nurturing the next generation receivers. Also, he received a totaling and impressive 1 crore fueled funds from ICMR, DST, DST SERP for their continued efforts to push the boundaries of glitazone research. So, with two very great impressive records, we are very honored to have Dr. Prashant Kumar sir from JSS College of Mysuru Pharmaceutical Chemistry Department to share his insights and expertise with us today. So we are very delighted to welcome you once again, sir. So I'm overing to you, sir. Please take the stage, sir.
Okay. Thank you very much, Madam Divya, Madam. I'm going to present today on glitazones and use of Discovery Studio tools for various biological applications in drug discovery process. Okay. Basically, I'm a medicinal chemist. So, as a medicinal chemistry, this presentation is all about the medicinal chemistry. In that medicinal chemistry, one important segment is the computational tools that we use it for the different applications. So that I'm going to focus it on. So it is a complete picture of medicinal chemistry, in that a segment of computational tools that are being used in this drug discovery and drug development process, like that. So at the outset, I'd like to thank Alltime Technologies for giving me this opportunity to present before you all. And I personally thank uh, Dr. Divya S. Madam. She is an application scientist in Alltime Technologies. So I wholeheartedly thank her, and also Sakshi Bharadwaj and Sheetal, who are accompanied her today for this presentation. Thank you. Now, let us look into the history of drug discovery and drug development process. In the allopathic system of medicines, that means what are the medicines that you can find it in the medical shop today. If you just look back that the way how they have been come to the market in the chronologically from early 19th century, that means around 1800, 1810, during that time, there were natural products used to be there in the market, like that. Okay, for example, like morphine, cocaine, they are from the natural source. So they were being used and sold in the market, like that. But what has happened in the same 19th century itself, by the late 19th century, there were very fewer natural products were there in the market, and more synthetic substances starts coming into the market. Synthetic substances means you all know that these are like small molecules, organic compounds. Okay, so these small molecules are started coming as a synthetic substances to the market like that. So if you, by the curiosity, if you look at the way how things have happened in the past, at that time, a dye and chemical making companies started doing the drug research and drug discovery applications like that. So today, if you look at one such company, is that Bayer. Bayer was basically a chemical and dye making company turned to do the drug research. Okay. Today it doesn't happens like that because it is a completely different setup what we require for a chemical company and the pharmaceutical company. And then slowly time passes away. In the 1905, in the early beginning of the 20th century, John Langley makes a very clear statement that a theory of receptive substances, saying that the whatever the drug that we take, it has to go and bind to the specific receptors in our body to elicit its desired pharmacological response or physiological response. So this becomes very clear that, so that means this will be the beginning for the rational drug discovery, rational drug design like that. So in that context only, in the 1909, first attempt was being made for the rational drug design. Okay, so rational drug design, that means hypothesis driven drug design. Okay, with a definite rationale for the drug discovery and drug development process. So the goal was that a safer treatment, and two important scientists, I'll name it, Paul Ehrlich and Zachariah Atta, they wanted to maximize the therapeutic index, a window that opens between the efficacy and the safety of the profile of the drug. And they wanted to maximize the therapeutic index, and they synthesized 600 compounds and evaluated their minimum curative dose and the maximum tolerated dose, and they found this salvason. This is the structure of an salvason, as you all can see it. Okay, this salvason we have used as an anti-infective drug till 1940, like that it was being used. Okay, you all know that what has happened in the 1940. Alexander Fleming discovers the penicillin, and penicillin replaces this salvason. But the question is, from 1910 to 1940, 1940, we have used this drug as an anti-infective drug. Okay, so my dear friends, closely look at the structure of salvarsan, which we have used it as a drug in the past. If you look at, okay, even though it looks like a dimer, but there are some arsenic atoms are there. Today we do the limit test for arsenic, okay, to ensure that it should be there in the negligible amounts, okay, in the pharmaceutical preparations. But we have used a drug which itself will contains the two atoms of the arsenic. Okay, so this has happened in the past like that. I am bringing it to your notice like that. This is what has happened. Next, in the 1960s, okay, first successful attempt to relate the chemical structure to the biological action quantitatively by the hands in the form of an QSR comes into the picture. So at that time, 1960, the IT sector was booming up. Okay, computers, hardware, softwares, and all a lot of developments which have been happening. So at that time, there was a hype that this computational tools, okay, that means using the computers, we can do wonders in the drug discovery and drug development process like that. Okay, so from the mid of the 20th century to the late 20th century, in the last century. Okay, so this was a golden era of this drug discovery and drug development process, as many of the drugs which are there till to, even now today in the medical shop, which we are all using it, they all are come from mid to the late 20th century, especially from 1950 to the 1970s. After the Second World War, So whatever that period which where lot of drugs which have come into the market and we are using it today. Okay, so that is the fine. But later on, this number of drugs coming to medicines coming into the market have been reduced, and we are running out of the fuel now. Okay, for especially for infectious diseases. Okay, to treat them, especially for tuberculosis and all those things like that. So, from mid to the late 20th century, understanding the disease states, biological structures, process, drug transport, distribution, metabolism, medicinal chemists use this knowledge to modify the chemical structure to influence the drugs activity with the advancement in the computers. This was happened in the mid to the late 20th century. Now, this is the how the drug discovery and drug development will be there. That means we start with identifying the disease. Disease is identified based on the symptoms. The symptoms are in turn connected with the biochemical pathways, and the biochemical pathways in turn will have an proteins in the form of a receptor or an enzyme, which will be which needs to be regulated to cure to find a treatment for that particular disease. So in that way, identifying the target protein, and then identifying the target validation, and then finding the hits hits to the lead and lead to the lead optimization, and then finally identifying the some candidate compounds for the preclinical testing, and then with the preclinical testing, if anything is very promising, then it comes to the clinical trials, and with the filing the investigation new drugs IND, and then clinical trials on phase 1, phase 2, phase 3, and then with the filing for the new drug applications, it will finally comes to the phase 4 of an clinical trial. So that will be in the market like that. So this is how the drug discovery and drug development will takes place, and simultaneously the formulation will also be thinking when the clinical trials are on the other way scale up is also happens like that. So these are all the things which will be happening as part of the drug discovery and drug development process take into the consideration. Now looking at the focus here, where a medicinal chemist will play an important role. So identifying the disease based on the symptoms, and identifying the what is the real biochemical pathway which is operating, uh, especially which is being affected in that particular individual to raise that particular disease, and that biochemical pathway we needs to be established, and what is the enzyme or the tar receptor we can keep it as a target so that we can do it. So in this process what happens is a lot of genomics work will be there, and proteomics work will be there, and potentially producing many targets, and personalized targets also will be the part of it. And a high throughput screening will be done when once you identify the target protein, and if you start producing the target protein by recombinant DNA technology. Okay, so then we can take that for the bioassays, especially for high throughput screening. In the high throughput screening, okay, we can screen millions of compounds per day, so that we'll get at the end of the day we'll get a data of which are all the hits which are hitting that particular target. This is what is the exercise what a pharma industry does it. Okay, so reputed pharmaceutical industries will be running an high throughput screening for these targets to identify the hits, and then here to assess these for that libraries of compounds are required. So how do we get them? So a pharma industry will also have a libraries of compounds. Okay, so that they it uses it for the screening for the high throughput screening like that. So to assess this libraries of compounds, one such technique is combinatorial chemistry, combichem techniques, where we can produce in different combinations and different permutations. We can produce large number of compounds in a quick time. Okay, and here the molecular modeling techniques, okay, computer graphics models that improve the activity, what modifications can be done. So here also the computation tools will also plays an important role. Okay, so in vitro and in silico ADME models, ADME predictions, all computational tools can be used it at this early part of the drug discovery and drug development process like that. Next, so this is the pipeline in the drug discovery and drug development process, especially from the medicinal chemistry perspective or pharmacologist perspective. So target identification to the taking it to the high throughput screening and then finding it out the hits and then hit to the lead and lead to the lead optimization to get the candidate compounds for the preclinical followed by clinical trials like that. Okay, so if this is how the drug discovery and drug development takes place, then what is this whole industry, whether it is an academics or industry or any agency, if you are involved in the drug discovery and drug development process, what for we are to work in the sense to discover the drugs. So here even academics or industry, everyone will be behind the two important things. One is the time and the money involved in the drug discovery and drug development process. So the time from the conception to the approval of any new drug is typically about 10 to 15 years of time. This is the average time it takes. Okay, so the estimated cost to bring a market drug to the market is that it is $1 billion, US dollars it is. So it is some 10 years back it was $1 billion we used to say. Now with the inflation, okay, it is almost $2 billion US dollars it will takes now. So in the 2021, now things have been changed with the advancements in the computational tools and all those things. Okay, so even it can be possible within 2 to 5 years of time also. Very good examples is like your vaccines which have been developed during the COVID-19 time. Okay, and also some anti-HIV drugs are also in that pipeline as well. So now, what drives this future of this drug discovery process? How we can reduce the time and the cost involved in the drug discovery and drug development process? Okay, how to how efficiently we can discover the drugs? Okay, that is hard to predict, but most of them say that techniques such as artificial intelligence, machine learning play a very important role. Okay, and in this context, what I say is that maybe the cost of the drug discovery and drug development instruments are being reduced so that everyone can start using these instruments. Okay, especially if you take plate readers, NMR, or HRMS, some big instruments and all. So they are very costly. HTS, if you take into consideration, for example, high throughput screening, it cannot be done. Usually, not nobody will does that academic setup in India. The reason is it is very costly like that in order to make it like. If these costs have been reduced, maybe we can see that many of them start doing the drug research and drug discoveries, and we may discover the drugs in the future like that. That is what I think like that. So, how drugs are being discovered? You believe it or not, many of the drug discoveries, if you take a penicillin drug discovered by Alexander Fleming, okay, it is by an accident. That means serendipity. Okay, so but what is the routine procedure? How we can do? How rationally? How hypothesis-driven drug discovery can be done? It can be done, okay, by screening of an new molecules. New molecules we need to screen them. Okay, these new molecules needs to be designed appropriately. If it has been designed through computational tools, it is more appropriate to use it and then screen it, okay, by in vitro and in vivo. That means screening by in silico first using an computational tools and then in vitro and then followed by an in vivo like that. So in order to get these molecules, you should have a synthetic lab also. Synthetic in the lab start synthesizing these molecules, gives it to the screening department in the pharmacology or physiology. So there they do the screening of these new molecules like that. So here, modification here. One thing is that there are two things into consideration: first in class and the best in class. When we are screening any new molecules, sometimes we notice that one of the hits, it may be the first in that particular class of an compounds. Okay, so it may be successful enough to go through the clinical trials and comes into the market. For example, if you take penicillin, so that is the first in class in the beta lactams, if we take into consideration. But whether that penicillin is being used nowadays, if you take into consideration, okay, so we are not using it that much. So the best in class, that means after that, okay, what are the beta lactams which are come to the market, which we are using it right now, the best in the class, like that. So first in class and the best in class, this the competition arises amongst the molecules in the drug discovery and drug development process. Next is the modification of existing drugs for the improvement. Always there will be competition for whatever the drugs which are there in the market. So there is always some kind of an demerits or disadvantages with the existing drugs in the market. So we'll always be looking for modification of that drug to find an, okay, to become more appropriate, to be useful as a drug for the take into that particular context into take into consideration. So mechanism based drug design, it is based on the mechanism of action of these drugs, which target it is intended, whether it is an agonist, antagonist, whether it is inhibition of the enzyme, what it is. Okay, so what needs to be done? So based on the mechanism of action of that particular drug, so we are trying to discover the drugs. Okay, and combination of these techniques, by combining one of these techniques, also we can discover the drugs, like that. And very importantly, the computational tools will play a very important role in this context of an drug discovery and drug development tools. Okay, so today we have computational tools and we can use them for discovering the drugs, like that. So drug discovery, I'm taking you now with this elaborated view on the drug discovery and drug development process. I will take you to one such case study where it is a case study on the glitazones. Okay, so drug discovery is one of the most important areas of research today because it is always, and there is a need to find a newer drugs for many of the diseases with advantages over the existing drugs in terms of potency, efficacy, and safety. Okay, so the ideal way to pursue this task is to design the ligand with the definite rationale for specific target and then synthesize it and screen it for the same. So in this context only, we uh, worked on glitazones, okay, as a case study, like that. So to give an introduction to these glitazones now, okay, if we take it, the causes of death, which is reported by NIH recently, so you can see that majority of the deaths are because of ischemic heart diseases, other heart diseases, and then the third one, it is diabetes, and next is the cancer, like that. So this diabetes becomes a silent killer. Okay, so due to which many people will die. So when diabetes first, any patient who enrolls for this disease, diabetes, so normally they'll be in the type 2 diabetes mellitus, and then they'll eventually move towards the type 1 diabetes mellitus, which is non-insulin diabetes mellitus to insulin dependent diabetes mellitus. Okay, so this is the trend. It is reported. Okay, that means the overall patients with this diabetes, the diagnosed type 2 diabetes are the majority here. Diagnosed type 1 diabetes are the small portion of it, and still there are undiagnosed diabetes taking across. This is across the world. Okay, many developing countries also take into consideration. This is the picture. That means many people have diabetes, but they may not be knowing that they have an diabetes. This is the picture it wants to depict here. So the incidence of diabetes is rising all over the world at fast pace. It's basically a metabolic disorder that results from the defects in both insulin action and in its secretion. And the commonly used hypoglycemic agents such as sulfonylureas known to stimulate the insulin secretion by the pancreatic beta cells, which often cause severe uncontrolled hypoglycemia, weight gain, etc. All these problems. So drugs which can reverse these insulin resistance without stimulating the insulin release from the beta cells can fulfill a major medical need in the treatment of non-insulin dependent diabetes mellitus. Like so, in that context, these are all the treatments which have been available today in the market, which acts through different routes like that. So, one thing which we are going to concentrate now is through these glitazones, which act on this adipose tissues for acting on the PPR hormone, PPR alpha receptors to enhance their glucose uptake and reducing the free fatty acid output and thereby reducing the insulin resistance, thereby enhancing the insulin sensitization as a treatment for the type 2 diabetes mellitus. As in the nutshell, if you want to say, reducing the secretory demands placed on the pancreatic beta cells by chronic insulin resistance can delay or prevent the onset of an type 2 diabetes mellitus. Okay, so that means that beta cell rest is an beta cell protection. That is what we can understand. So we can, rather than the beta cell is getting exhausted, we are trying to give a rest to the beta cells. Thereby, its beta cell has been protected like that. So by reducing the insulin resistance and enhancing the insulin sensitization by the receptors like that. So now thiazolidines or glitazones, the mode of action. What is their mechanism of action? The mechanism of action, they act on PPR gamma. So this PPR gamma basically takes care of the carbohydrate metabolism. Okay, the enzymes which are being required for the metabolism of these carbohydrates in the physiological system. So that will be driven by this PPR gamma receptors. Whereas PPR alpha, it takes part in the lipid metabolism. Okay, like that. So these thiazolidines are otherwise called as glitazones. They basically act on PPR gamma or PPR alpha. The relative extent it will depends upon one glitazones to another glitazones. Some glitazones, but majority of these glitazones act more on PPR gamma receptors. To some extent, it will act on the PPR alpha receptors. I'll tell you the reason why it acts on the PPR alpha receptors also. But before that, let us look into this mechanism action of these glitazones now. So these thiazolidine ions of the glitazones has to cross the cell membrane, and through the cytoplasm they have to reach the nucleus, and they have to cross inside this nuclear membrane to where they bind to this PPR gamma receptors. These PPR gamma receptors will be there in augment with this retinoid X receptors in complex with the retinoid X receptors. So that is why this PPR gammas also will play a role in the inflammatory processes because of their association with the retinoid X receptors. Like. So this complex then further it will it will finds the transcription factor which is getting signaled by the when the insulin binds to its receptor, the post insulin receptor binding signal through the transcription factors, they get activated, okay, in the presence of an this PPR gamma retinoid X receptor complex, which is activated by thiazolidine because of its agonistic action, then followed by complementary DNA to the RNA, and then the protein synthesis for the synthesizing the enzymes which are involved in the carbohydrate metabolism will be taking place through downward signaling process like that. So this is how the mechanism of action, if it is being from the thiazolidine ions or glitazones will be acting. But normally these PPR receptors, peroxisome proliferating activator receptors, are generally referred as an orphan receptors. Orphan receptor means their natural ligand for their which binds to these PPR gamma receptors is still unknown. Okay, any receptors which exist in our body should have a natural agonist in the body, so that is not being known. Like, so drugs from this class of glitazones now, if we take into consideration, this is the troglitazone, this is the drug troglitazone. So this is the first drug which comes into the market in the 1997. Okay, so at that time we termed these glitazones as a novel class of anti-diabetic agents. Okay, like that till if you refer Goodman and Gilman textbook, it is a novel class of anti-diabetic agents only. It classifies it. Okay, so now what has happened is this medication was termed as an so what has happened in the January 1997, Parker Davis company in the form of Resolin, it comes into the market. But in the December 1997, by the end of the same year, okay, it was been recalled from the market. It was been withdrawn from the market. Okay, so because of its concerns of hepatotoxicity, it is a major concern. Some deaths were also happens because of this. So because of that, it has been withdrawn from the market. So it is very hard to digest for a pharmaceutical company having been invested so much of money in this current drug development process, and within a year it has been recalled from the market. It was okay. So now, if you look at the way how these glitazones we could able to discover it, okay, it starts from fibrates. Fibrates, you know that it is a lipid-lowering agents, okay. So this clofibrate, if it this is the structure of clofibrate, if you look at this has been derived and fibric acid made into carboxylic acid here, acidic head. Then this fibric acid has been converted to the derivative of this fibric acid like this, and then this fibric acid as an moiety of this is the acidic head, a carboxylic group, and then the hydrophobic trunk, and then the linker here, and then the hydrophobic tail. So this was the basic structural requirements for these class of compounds, and with that we could able to develop the cyclitazone, troglitazone, pyglitazone, rosiglitazone like that. Once this troglitazone came to the market and it was being withdrawn in the 1997, and pyglitazone it was there 1999 it was being introduced, and still till now it is till today it is there pyglitazone. Okay, so rosiglitazone was there from 2000 to 2010, almost 10 years it was been there. Then it was been withdrawn from the market because of its cardiovascular risk. So if you look at here, these fibrates, they are more lipid lowering in nature. But if you look at the structure of these glitazones, if you look at here, they are less lipid lowering in nature, like that. So if you look at on the other way, in terms of carbohydrate metabolism, less glucose lowering in nature. Okay, and but these glitazones are more glucose lowering in nature. That is depends on what is their mechanism of action. Okay, so these fibrates acts on ppr alpha more, so that is why they tend to be in lipid lowering. Whereas if you take these uh, glitazones, they act on ppr gamma and tend to make it more carbohydrate metabolism, they take part, and tend to reduce the glucose lowering by the insulin, by reducing the insulin resistance and enhancing the insulin sensitization line. So, saraglitazole is this is the drug which is developed from India, and to be honest, this is the first drug from India which has been okay developed indigenously developed in India. Okay, this was from Zydus Cadila. So this Zydus group has developed this saraglitazole, and it is used for the treatment of type 2 diabetes mellitus from this class. So it is finding its nod for US FDA, so that clearance it is still has to get it slow. So, what is the rationale for the further development? Despite many advantages, there are several factors that limit the use of thiazolidine ions or glitazones. So, the major factors are numerous side effects associated with these glitazones, apart from hepatotoxicity and congestive heart failure and cardiovascular risks associated with these glitazones. So, the focus today is therefore on the development of novel PPR gamma agonists, which might surmount the problems associated with the known glitazones and offer advantages as an anti-diabetic agents. Therefore, we have made an attempt to design this synthesize some evaluate some non-glutazones. So that case study only I will be discussing with you. So now how we designed our plans. Okay, so we computationally with the pharmacophore models using a training set and molecules, we best mapped molecules have been taken further from this pharmacophore model of discovery. We have used Discovery Studio for this purpose, and from this through the pharmacophore modeling and pharmacophore based design, based on this mapping which has been done with this pharmacophore based design. So then we have subjected it to the in silico admin studies. Okay. Again, we have used admin properties of this Discover Studio and TopCat. We have used it, and we identify the admin properties of these glitazones, what we have designed it. And then we have done the conformational analysis, molecular docking, and simulation using the Discover Studio tools. And then we, whatever the designed molecules which are there, that we have been taken it for the synthesis. Okay, so we synthesized it and we characterized it, purified it, and analyzed it, and then taken it for the in vitro PPR gamma binding assays, and then followed by cell based assays, and then followed by in vivo assays. So these are all the things which we did it as part of the this drug discovery and drug development project uh, with the aid of my PhD scholars. Actually, my PhD scholars, Prabita, Yuvraj, Shubhankar, and Fayaz and Priya Durai. So they're all are doing the research. Okay, so so on behalf of them, I am presenting it now. Okay, so now when we did the initial studies, we came to know that these glitazones needs when these kind of an interactions like that based on the literature survey if you take into consideration. So this histidine 449, histidine 323, and tyrosine 473, glycine 286, cysteine 285, methionine 364 and 329. These are all the very important interactions for these glitazones to exert that its physiological or pharmacological actions like that. So in that context, also we understood that based on the literatures, these are all the structural requirements which have been required. That is thiazolidine dione as an end, followed by an benzyl oxy as an trunk, a benzyl oxy group, followed by two carbon linker, followed by an hydrophobic tail. So this is the structural requirements for these like. So in that process, in the after this pharmacophore mapping and pharmacophore modeling, okay, so we subjected these molecules for the in silico admit studies using Discover Studio, and you can see that majority of them, some outliers of this uh, have been removed, and what is coming within these inside this admits permissible areas, they have been taken into the next stages of a drug discovery and drug development process like that. They have been taken further. That means next we calculated their admits and properties, for example like c log p, and log p which plays a very important role in the absorption of the drug, okay, and also in the its distribution, and then log of solubility is been taken into consideration, and topological polar surface area has been calculated, and then human intestinal absorption HIA has been calculated, and then plasma protein binding, blood brain barrier, and acute oral toxicity. See, if you take acute oral toxicity, to do this, you need an animal experimentation, you need an animal ethics clearance, and then you have to do this experiment. And before that itself, just by the click of a button using this Discover Studio, we can able to calculate these all these parameters very easily, very comfortably. So LD50 is also it is there, and later on we was matched with in vivo results also. It was quite correlating well with the in silico results. That means that what are the Discover Studio tools are there to predict these handmade properties, they are good enough like that. So to continue with this case study, we are taking a couple of molecules, some series of molecules we are taken it, and we have next after this admit studies, it has been taken it to the um, this docking, and in the docking we have used initially we have used a lid dock, and then we have used the sea docker, uh, and the sea docker, it is predicting better, we have observed it. Okay, so that this is the C docker energy, and this is the C docker interaction energy, which it says that for the all the interactions that it makes a ligand with the its PPR gamma receptors, then this is the energy that it has been the case. So that has been when it has been compared with the IC50 values, when which will be has been calculated by in vitro experiment. Okay, so it was quite matching with the in silico experiments. Okay, so this interaction energy or the C Docker energy, it was matching very well like that. Quite correlation has been observed. And the interactions, if you take tyrosine 473, whatever the interactions I wanted, I have ensured it by the docking. And these with these interactions, I issued in 449, issued in 323. Okay, so with these interactions only, I could able to proceed with the this thing. So here, what happens is this is the where we are started with these acting on PPR gamma receptors for diabetes actually, and slowly we came to know that these PPR gamma receptors, that means these glitazones, can also be a neuroprotective in nature. For its neuroprotectivity, this PPR gamma will be there in complex with the PGC1 alpha. Okay, so this PGC1 alpha activation through PPR gamma, once the glitazones binds to the PPR gamma, causes conformational changes in its receptor. That conformational changes in the receptor will causes some activation of this PGC1 alpha at the PGC1 binding domain of the PPR gamma receptors. So with that taking it as an advantage, we designed some glitazones for neurodegenerative disorders also, and we found that a very good results. That means glutazones binds to the receptor, causes conformational change. That change in conformation will be resulted in the conformational changes in the PGC1 alpha. So this is what we studied it, and we found it a very interesting case study using the Discover Studio C Docker multi docking tool. Okay, and then and also we have done the multidynamic simulations also, and these are all the cases with different drugs taking into the consideration how they are interacting with the PGC1, this PPR gamma, and the PPR gamma, how it was interacting with the PGC1 alpha taken into the consideration. So this is also a depiction of how the PGC1 alpha was being activated by the PPR gamma receptors like that. So there is an option in the Discover Studio to do protein-protein docking also. We can do PGC1 alpha with the protein also, we can do it. That option is also there. So now these are all the interactions which have been seen in these cases like that. So you can see at different distances the hydrogen bond distances, pi bonds, okay, hydrophobic interactions, dipole-dipole interactions, and hydrogen bonds, all these things take into the consideration like that. So now if you take to know that whatever this the rosiglitazone which we docked it, okay, so we processed it in a similar way, and uh, done the docking, and then we checked with the co-crystal ligand of this rosiglitazone, okay, and it was found to be very close, not much of a differences in that. So that ensured that whatever the Discover Studio tools that we are using it, they are quite accurate enough to predict it, the binding mode for these ligands to bind at the PPR gamma receptors like that. Okay, so this is the And then what we did is we started synthesizing these molecules. So while synthesizing, we have kept three things into the mind. One is the hydrophobic, this acidic head, and then followed by a hydrophobic trunk, followed by a two carbon chain linker, and then the hydrophobic tail. Like that, take into consideration, and we designed a libraries of compounds, and these libraries of compounds we have been designed, and it has been synthesized in the synthetic chemistry laboratory, and then it has been purified and analyzed and then checked for its activity, like that. So this is all the developments which have been taken place now. So everywhere for developing the synthetic schemes also we play around. But what happens in the a real as a medicinal chemist, I wanted to tell you is that we may predict whatever the compound, we may design whatever it may be, but its synthetic feasibility is a big question. Take into the consideration that what facility we have. Okay, in our laboratory, what kind of reactions we can do it. Okay, so what kind of a facility that we have. Okay, and take into the synthetic schemes, and what can be done. For example, if you take a hydrogenator, sometimes some laboratories may not have an hydrogenator, so then if you are planning for any reduction like that, it may be a difficult. Okay, if it involves like an palladium charcoal with hydrogen passing gas, like that. So it all depends on the your setup of your synthetic chemistry lab setup, so that it to take up this design libraries of compounds for the synthesis, like that. So this is the, in conclusion, what I want to say is that docking and dynamic can notably noticed specified that potential ability of the synthesized compounds to active towards the neuroinflammatory conditions, as well as we have explored nearly about 500 molecules we have synthesized, and we have checked for its anti-diabetic conditions also. That means as an anti-diabetic agents also we have screened it, and also recently we have screened it for neuroinflammatory conditions also, like that. So these were all the publications which we have published it in the recently, like this. So coming to the synthesis, how we can plan it. So you can plan it from the right from the back, from that means from the tail portion. So we take an aromatic amines, treat it with chloroacetyl chloride. This happens to be the two carbon linker, and then connect it with the okay, the acyl amine portion where it gets connected. So this is an acelamine, and then this has been connected to the vanillin. Vanillin is an aldehyde, an hydrophobic trunk. It will becomes like that. Vanillin is a natural substrate. It is natural product. So that if we can use it as a building block, it is always beneficial. Then we can use this. Then we can condense it onto the thiazolidinedione, like that, and it will becomes like an acidic head, like that. Then we can reduce it to become it like a single bond, like that we can make it. So how can we generate this thiazolidinedione? So we can take this chloroacetic acid, treat it with thiourea, and then it will form some 2-aminothiazolidine 4 on a cardiac precipitate, which then heated with HCl, it will get cyclized to form thiazolidine 2,4 very stable ring, like. See, another modification is that we can take this vanillin and we can connect it with a two carbon linker, 1 to dibromoethane, we can connect it, and then we can connect it to azide and we can subject it to click chemistry, like that. Another way is connect it to another phenol, and then we can make some derivatives out of that. So in this way, it is very interesting in the as part of the synthesis to generate libraries of molecules, like that. One should have interest in working in the synthetic chemistry lab, so then it will be really interesting. So like this we have taken this benzaldehyde and we have made into dimer, and then we are condensed onto the thiamine dimer, and then with different derivatives and to form different derivatives, like that. Another way is we have taken this started with simple glycine, and then this glycine has been protected, the C terminal, N terminal of it has been protected with the bulk, and then the C terminal is extended with connection to the phenolic group of this. vanillin, okay, through 1,2-dibromoethane, and then it has been condensed onto the thiazolidine dione and then reduced it to become a thiazolidine dione. So whenever we do any synthesis, we need to confirm it for the structures. How do we confirm it? We characterize it with the conformal structures. So this is the thiazolidine. How do we say that thiazolidine? Because it has a mass of 117. So the M minus one peak which has come here at 116, like that. Okay. So higher NMR will confirm the peaks. So this is the NMR confirmation of these glitazones, like this. So very interesting thing is that if you can see this double bond CH here, it comes at 7.62, okay, delta ppm, like that. If it comes 7.62 like that, then there will be an indication that this could be the possibly the possibly because of this intramolecular interaction which is possible, thereby it will becomes like a Z configuration. This is a Z configuration, a geometrical isomers. So as part of synthesis, you need to play around with respect to the stereochemistry of the molecules also. So those things will be there. So next, this is a heterocausic, where in the heterocausic we get an cross peaks inside this square, where these peaks has been connected to the carbon and the hydrogen. If you horizontal lines, if you draw it for these horizontal and vertical lines, you can see which carbon is connected to which hydrogen, like that. Okay. So in that way only we came to know that this double bond CH, which normally comes between 5 to 6, it has come at 7.62, like that. And with that we came up with this confirmation for this particular um, uh, geometrical isomer. It is a Z isomer, like that. Okay, Z configuration. So these are all the batch of glitazones which have been produced by us. Okay, so in the batches we did this production. So at the every time whenever we have used these computational tools, okay, especially like drug discovery. As a Discover Studio tools, what we have used it. So this is in the pharmacophore model one case study. If we take in consideration the predicted activities and the actual activities, what we have found it in silico, it was found to be 0.813. That means 81% accuracy is been there with the prediction of these Discover Studio tools. Okay, that is what we can say it. We have used this Discover Studio extensively in the past 10 years. Okay, and we have found that they are extremely useful. Okay, so uh, I have a great pleasure in recommending this Discover Studio tools, because we are using this Discover Studio tools. You can do the ligand-based drug design, you can do the structure-based drug design. As part of the structure-based, you can do the de novo drug design, docking. Okay, so structure-based pharmacophore. As part of the ligand-based, you can do the 3D QSCR. Okay, pharmacophore models. So and ADMET studies. Okay, so all these tools are enriched in this Discover Studio, and we have used it and very efficiently. And this Discover Studio is also quite cost effective to afford it in our laboratories also, and that is why we purchased it and we have used it in our laboratory, like that. So finally, the SAR structure activity relationships we need to we derived it for these zones. What is the requirements? All those things we have derived it, and then for any work there will be in some future work suggest what all the future work we can able to do it. So this are all the future work suggest. That means that we are trying to redesign these glitazones using Discover Studio tools even right now in our laboratory, and we are repurposing these glitazones apart from diabetes to the neurodegenerative diseases like Parkinson's diseases and Alzheimer's disease, like that. So those projects are also ongoing. Okay, so some of the schemes that I told you, okay, so that can also be further extended. If you are also in interested in working in these glitazones, you can also work on, because there is a lot of room for the improvement here. And you have, either you can take it to repurpose it for the neurodegenerative disorders like Parkinson's disease or Alzheimer's disease also, and make it useful like that. And I'd like to thank. This is all about my this, and then I'd like to thank Divya S. Madam. She was very supportive in the. Apart from providing this Discovery Studio tools to us, they provided a back-end support in uh, whatever the problems we had, issues where it was being solved by them, with our PhD scholars and regular seminars, workshops, webinars, and all we are conducted in that process. They are all very fruitful and very helpful for us to grow all together. So thank you very much. Thank you.
Yeah, thank you so much, Dr. B.R. Prashant sir. It's an insightful presentation about the beta zones. Certainly, I don't think so. No one can explain as deep as possible, covering all the schemes. So quickly, we can go for an Q&A session. So,
Okay.
can you just post a question? Yeah, thank you so much. The question is, you can see it on the screen, sir. How do you study, admit, and hepatotoxicity at the earlier stages to prevent failures?
Yes, yes, definitely. It is a valuable question. Hepatotoxicity is one of the major concerns for the drug discovery and drug development process. As we can see, this troglitazone, one of the glitazone, which was been from the Parker Davis company, it was been released in the market in 1997, January. But by the December 1997, it was been withdrawn because of drug-induced hepatotoxicity concerns like that. So, as a trial from my side, as a medicinal chemist, we checked it, the troglitazone structure in the ADMET studies, and it was showing the hepatotoxicity in the ADMET profile in the top cat. It was showing it. So, similarly, we studied our glitazones also for the hepatotoxicity and ensure that this hepatotoxicity was been not a concern for these glitazones. Some of the glitazones that we had constructed, there was an issue, so we dropped those glitazones for the synthesis and evaluation, and those glitazones which didn't show the concerns of hepatotoxicity, they only will be taken into the consideration. And also we have top two compounds out of these batches. We have taken it for the in vivo studies also, acute toxicity, and followed by chronic toxicity studies according to AAC's guidelines. And then we found that it was passing these initial studies in the in vivo. That means that whatever the in the Discover Studio tools, whatever the tools which are there, they are quite accurate enough to predict the admit studies in the beginning itself, like that, including this hepatotoxicity taken into the consideration.
Thank you so much, sir. Can you pass next question? Yeah. So,
Yes,
can
ma'am.
we study protein-protein interactions? I think
Yes, yes,
sir is
ma'am.
he is answered. It seems.
Yes, yes, that can be studied. Protein-protein interactions can be studied in Discover Studio, no doubt about that. We have studied it also. Between PPR gamma and PG7 alpha, we studied protein-protein interactions. Okay, that I have explained it in my presentation also. So that can be studied. That's not a problem. It is very comfortable in the Discover Studio tools. We can study it.
Okay, there is another question. So, how pharmacophore modeling of glitazones bolster the synthesis using discovery studio?
Pharmacophore, because these glitazones, if you take into consideration, they are like, if you consider it like a bird-like structure. So it has an head, trunk, followed by a two-carbon linker, followed by an hydrophobic tail, like that. These are the structural components of an glitazones. It has an acidic head, then hydrophobic trunk, two carbon link, and then the hydrophobic tail. So using these structure activity relationships, okay, these pharmacophore features, that helps you, that guides you from the literature point of view also, and also in the pharmacophore modeling also, it helped us to screen the molecules uh, based on the pharmacophore based design molecules. So in that way also it will be very helpful like that. So yeah, it is pharmacophore is well established in the case of an glitazones, that you can make use as part of the structure based pharmacophore or ligand based pharmacophore modeling, and we can develop further glitazones, and we can see that synthetic feasibility, and then we can synthesize it in the lab, and we can check it for the activity, and we can revert it like that. So this kind of an exercise we are trying to do it, and we are quite successful in that way in doing that.
Yeah, thank you, sir. And there is one question: how to choose a PhD? It may be a very generalized one. How to choose a PhD topic which combines a dry and as well as a wet lab?
It is very simple actually to choose a PhD topic. It is one thing is that I choose this glitazones because when I was doing my masters, at that time my supervisor told me that you come up with some problem like that. So I went back to my home, and I thought of what can be done like that. So I was new to this research at that time. So then it was in my mind that see, my parents, they are diabetic, they have diabetes, hypertension. So I had to do something for them. So I thought that why can't we do some research on diabetes like that? So that made me to take up something related to the diabetes like that, these glitazones like that. So this is what has happened in my case, I'm telling you. But for you as an youngster, I would like to guide you is that very simple, actually, no need to think very complicated, but still, you have to make an effort to do some literature survey. So, to be a writer, you should be a reader first. Okay, so in that context, what happens is you should read lot many articles. Okay, so you should see the articles written in some reputed journals, what kind of an work they have been presented. Try to understand them. So if you try to understand what others are doing the research like that, then you can, then only you can plan your research in the similar way like that. So we should be good at observing what others are reporting. We should understand that, and then based on that, we should see what best I can do it in this case, like that. So you can take up any problem for that matter, not necessarily on the reasons only. Any problem, PhD, you can topic, you can choose it, but you should have that passion to explore things further like that. So that is very important. Yeah, your guide can also assist you, your supervisor, if you are little not to that level of understanding, then your supervisor should lift you you up. So in that way, both the student and literature, because we see that some of the students, the PhD scholars who enroll for PhD, some are very brilliant will be there, but some are little poor also will be there in understanding the things like. But still, if you make things conducive for those students, scholars, so they come up very strongly and they do extremely well. So all these things we have seen it in the as part of the PhD programs like that. Some so hope you will also take up a topic which suits your facility. What is your facility? What kind of research can be taken? It all depends on your facility where you are working, what facility you have, what kind of, and whether you have a computational tools, you have a synthetic lab, you have an analysis lab, okay, you have a pharmacology lab, you have to see what is their coordination, everything else, and from other departments. So based on that, you can decide what kind of a topic it will be taken into the consideration. Like,
Yeah, thank you so much for motivating the students, sir. So we have another question. Sir, can we generate QSCR model to predict the activity of unknown compounds?
Yes, surely. Why not? You can say in the QSR, what happens is that we generate a model using the training set. Training set means which are uh, known compounds which are for the desired biological activity. We'll take it in a diverse range in terms of structural diversity, so and in terms of potency. So next, then we develop a model. That model can be used to predict the activity of an unknown molecules. Any unknown molecule in the developed model, if you bring it, we can predict what is its activity like that. So this test set, it is nothing but the external set. It is not taken in the development of that model, so that can be bought into this developed model, and it can be predicted for its activity like that. So any compound which is yet to be synthesized, it not have an existence at all. It is yet to be synthesized. That can be checked for its activity using the these QSR techniques like that. So we have also done QSR techniques using this Discover Studio tools. So Divya madam has also helped us in that process, and we could able to do the QSR studies also. Like,
Yeah,
thank you.
thank you
Definitely,
so
we
much, sir.
can.
Yeah, sir. And there is another question: how to validate the docking study?
How to validate the docking study? As I said you, I showed you in my previous slides, is that like if you take this. This is the one. Okay, fine. For example, this is the co-crystal ligand and the what the dopped ligand, if you take into consideration. Okay, if you superimpose it and if you find out the what is the RMSD, the RMSD should not be more than 2 angstroms. Okay, so that is the consideration. So that is how it can be done. But in the real sense, if I want to say it, that means that in the real way of in doing the docking protocol validation, what we do is we sketch the molecule of an glitazone, we minimize its energy, and then we dock it, and then we check it that confirmation and with that of the co-crystal ligand confirmation. That should not exceed more than 2 angstroms. We say 2 angstroms, sometimes it may go up to 2.53 also. That is also quite acceptable. But this is how we do the validation of the docking protocol, whether it is correct or where
Yeah,
to ensure
right.
that whether the right.
So, any other question? Sir, how do you think glitazones can be used in neurodegenerative diseases?
Yes, because what happens is if you look at its process now, if you think here, what happens is see in this mechanism, what happens is that glitazones binds to PPR gamma. It will be there in complex with retinoid X receptors. So here it plays a role in the inflammatory pathways also. So in this inflammatory pathways, that PGC1 also will binds to this complex. Okay, so PGC1 alpha will plays a very important role in the neurodegenerative disorders, in the sense that it by activation of PGC1 alpha increases this mitochondrial biogenesis. If the mitochondrial biogenesis increases, that corrects the neurodegeneration. Okay, so it is like a neuroprotective effect. We can observe it. So then, in that context only, these thiazolidine diones, why we started using it against the neurodegenerative disorders. Apart from this insulin sensitization. Okay, that's it. So in that way also it can be explored very well. This for neurodegeneration, we have found very good results now. There is a lot of literature backup is also there for the glitazones to usefulness in the neurodegenerative disorders also. A lot of literature backup is also there. Apart from that, we have also found a very interesting results. Like we need to optimize, fine tune the glitazones to be very effective against these neurodegenerative disorders. That's what exactly we are trying to do it right now.
Thank you so much, sir, for things. And then there is one more last question. How BioVia Discovery Studio will support modern computational drug discovery for organic chemistry?
This BioVia Discovery Studio supports organic chemistry at various ranges in the sense if you take basic energy minimization, conformational analysis, okay, whatever the tools that we do it as part of the organic chemistry take into the consideration. All this can be performed using in Discovery Studio. For an organic chemistry, if you are an organic chemist, you can do wonders with these Discovery Studio tools. Okay, especially since you know that how the organic molecule looks like and how that shape of the molecule, how the conformation of that molecule you can able to predict means. It becomes very valuable tool this Discovery Studio for an organic chemist like that. Okay, so if you are an organic chemist, you can do get an inclination towards medicinal chemistry also, and you can use Discover Studio also, and it will be very helpful for you. For an organic chemist especially, since you are an organic chemist, you know synthesis well. So what are the molecules that you are designing it using Discover Studio, you can guess it at the stage itself whether I can synthesize it in my lab or not. That also you can think it. That is the advantage with the organic chemist like that. So for organic chemistry, you can suit this Discover Studio very well, and you can use it very effectively in your this thing.
Thank you so much, sir. I think everyone is appreciated. It's a very nice and informative session. Certainly, there is no doubt in this. So it was a great pleasure to join with you. I think it's a time for a conclusion. And in a nutshell, Alton Technologies come to platform. It's lovely to thank you, the distinguished speaker, who is a leading expert in the glitazones targeting PPR gamma with a profound impact on neurological, inflammatory, and diabetic research. Sir also has shown the significant influence of C Docker and its correlating with the biological IC50 value and the QSR correlation of about an 81 percentage and further deep diving and showing the understanding of a molecular dynamic conformational changes and its revolutioning in the drug discovery process. Additionally, the sir also explained the detailed scheme of compound synthesizers have enriched our understanding in the field. And this is a platform we like to thank a remarkable and distinguishable speaker, Dr. B.R. Prashant sir, for taking a great time in respect of his busy schedule, for sharing his great knowledge of zones with the people in the Altam platform. So thank you once again, sir.
Very much.
And yeah, we'll be definitely sure that all the participants who will attend across the country, this Altum platform will serve as a catalyst for new ideas, collaborations, and advancements in the computational biology and chemistry. Especially today, we had a great speaker in a medicinal chemist. Together, let's drive this progress together further and bring a positive change in the world of medicine. With this, I concluded and I once again thank Dr. B.R. Prashant sir for having us in a busy schedule. Thank you once again, sir.
Thank you very much.
Thank you, sir. Thank you, sir.
Thank you, Diva madam. Thank you very much.
Yeah, thank you. So, thank you, participants, for joining us. Stay tuned with us. We'll be coming back with the new interesting topics in future. Thank you, everyone.